Coronavirus, using your DNA against you. This is an independent research study where I used many sources of information, including NIH databases, to recognize something curious about the coronavirus and human DNA, and possibly how it could be doing its nasty stuff. To really fix something, you have to understand how it works, and that's what this video is all about. Here's what I found. There are hundreds of coronaviruses. Here you see the 7 that infect humans. The older ones, NL63, 229E, OC43, and HKU1, all seem to us like the common cold. A new coronavirus that jumped to humans from dogs appears to act like the common cold types. SARS and MERS are much more serious, but you have to be in close contact with an infected person, making them very rare. Wuhan CoV, otherwise known as COVID-19 and also very serious, transmits through the air, is easy to catch, and has caused a worldwide pandemic. We'll look at all of them to see why some are so much more serious than others. You can see that coronaviruses are basically round with spikes that inject the virus into cells. Here's how all the coronaviruses work. After the virus enters the cell, it unwinds and is fully copied. Then it can try to enter the cell nucleus or be coated in a protein envelope and released to infect other cells. Entering the nucleus would help the virus improve its infection agenda, but that's where our DNA is, and the cell makes it very hard to enter. I wondered if there was any clear link between the coronaviruses and humans. Since coronaviruses are RNA-based and humans are DNA-based, the link couldn't be direct, but I started looking at the nucleotides that make up RNA and DNA to see if anything popped out. The coronavirus has about 30,000 nucleotides, while the human genome has over 3 billion. I decided that a 1 in a million chance of a nucleotide string appearing in both the coronavirus and the human DNA would be good enough. DNA and RNA are made from 4 different nucleotides, so a string of 10 would occur 1 chance in a million at random, and I looked for a string at least that long. And I found these 3 nucleotide sequences in COVID-19. The 4 nucleotides in DNA and RNA are adenine, cytosine, guanine, and thymine. They combine to form everything we are. Those sequences happen to be DHU loops in an important cell component called tRNA, used for making proteins. Here's a 2-dimensional drawing of it. You see 3 loops separated by tightly bound stems. The virus doesn't have a tRNA. It only mimics the loops, and it doesn't have those tightly bound stems. Unfortunately, MERS, SARS, and SARS-CoV-2, which is the official name for COVID-19, don't have the right codes in the virus where the anticodon loop should be in a real tRNA. So the DHU loop doesn't appear to help the virus enter a cell nucleus. After a few beers, I decided to check the T loop, which only has 9 nucleotides, for double matches too, starting with COVID-19. Turns out it has a nucleotide sequence for the amino acid lysine. SARS also has a T-loop mimic for the amino acid lysine, the same code used by COVID-19. But since SARS is older, COVID-19 probably borrowed it from SARS. It turns out that the amino acid lysine is especially needed by the cell when it reproduces. Could that be why SARS and COVID-19 are more infectious than MERS? I also found another T-loop match for the amino acid valine, only in the Omicron variants. Omicron is much more infectious. Could this extra loop be a reason? And the anticodon for the valine tRNA was found in the SARS-CoV-2 just where it should be if this was a real tRNA. So, all SARS-CoV-2 varieties mimic one T-loop for the amino acid lysine, and the Omicron variants mimic yet another T-loop for the amino acid valine. Why does the virus go to that much trouble to arrange its nucleotides to pretend to be human tRNAs? MERS has a T-loop match for the amino acid tyrosine with the correct anticodon code in the right place to pretend to be a tRNA for tyrosine. If that amino acid is needed in the nucleus, that tRNA, or that virus part pretending to be a tRNA, will be admitted. Coronaviruses without those double matches won't get into the nucleus and will just have copies made. When an admitted virus associates with our DNA, it could become even more aggressive, able to use features in the nucleus to cause more trouble. If it suppresses our immune system, other diseases could jump in. We've seen that happen with MERS, SARS, and COVID-19. For simplicity, let's just look at what we can do about COVID-19, but recognize that the same approach will work with both MERS and SARS. We've developed vaccines for COVID-19 that stop the spikes from connecting with our cells, but don't attack the actual virus. Viruses are always mutating, and an easy mutation is to make different spikes that our vaccines can't stop. If the speculation that COVID-19 enters the cell nucleus and associates its RNA with our DNA is correct, then perhaps eliminating that process would make COVID-19 a relatively mild disease. This method goes after the virus, not the spikes in its shell. As you can see, SARS-CoV-2 has two different troublesome nucleotide sequences. I suggest the experimenter zaps them both. Then, using cloned animals to minimize any biological differences, the original virus and the zapped virus are tested. Hopefully, the original virus will cause a much more serious illness than the zapped one. After the healthier animals fully recover, they're infected with the original virus. Positive results would mean a personal vaccine for each individual has been created. In that case, we have a vaccine which must be tested on humans for effectiveness and to find and eliminate any horrible side effects. Then we've done it. We have a vaccine to stop us from dying by COVID-19, and our excellent biochemists and virologists can help us recover from illnesses the coronavirus is still able to produce. So, in summary, the large family of coronaviruses includes 8 that can infect us, with MERS, SARS, SARS-CoV-2, and Omicron being deadly. They can make our cells believe they're really carrying an amino acid needed in a nucleus. Then they associate with our DNA, causing all kinds of problems. I also suggest that SARS, SARS-CoV-2, and Omicron are the same disease that has evolved. A way to create possible vaccines for them was suggested. If you want to find out more, I've written a paper that includes all you've seen and heard, along with the data found, references, and a lot more detail. That's the story of my independent research on the coronavirus. But let's chat for a minute on independent research in general. Are you a free thinker with a novel and exciting idea that you want to develop? If it's a science idea, you'll find lots of information and data available, but you have to look for it. You should also realize that you'll be spending an awful lot of time alone researching your idea, with an awful lot of ideas being wrong, perhaps including my idea you just saw. And you're not getting paid. You have to really believe in your idea and in yourself to have any chance of success. If you do find something, you can certainly be proud of increasing world knowledge, but be careful how you express your findings. Try to use words like perhaps or speculate rather than prove or known, and have data around to back you up. Keep in mind that even Darwin's theory of evolution and Einstein's theory of general relativity are still called theories and not facts. It also helps to have someone who inspires you. In my case, it's my daughter Heidi, who was stricken with a brainstem tumor, but who continued helping troubled teenagers while becoming progressively paralyzed. Here's what she told CBS News.
I want people to know that they can do whatever they want, no matter what the situation is. They can do it. They should